Good morning, everyone, and happy Fossil Friday. I'm Brittany Stoneberg, the Outreach and Communications Coordinator for the Western Science Center. And with me, as always, is Gabe Santos.
Good morning, everybody. How are y'all doing on this wonderful Friday?
Good Friday. Gabe is the Outreach and Communications Coordinator and Collections Manager for the ALF Museum, as I'm sure you all will know by now.
Did I
And
forget to
joining,
introduce myself again?
I think you did.
I
It's
always
it's
do
Friday.
that.
It's Friday morning. We haven't had our coffee yet. We were talking beforehand about how neither of us had had breakfast. So this is what you get, everyone. And what you also get today is our guest today, Dr. Natalia López Carranza. Hello, Natalia. How you doing?
Hey, I'm good. How are you guys?
Good. I hope for your sake you have had breakfast, unlike Gabe and me here.
Yeah, it's a bit later here.
Yeah, so today Dr. Carranza is going to tell us all about something like I'm sure we're as paleontologists familiar with, but I don't know a lot about them. Brachiopods. So I am excited. We are continuing the chain of invertebrate paleontologists.
Inverts rule.
Yes,
Yeah.
all hail the inverts. So, a little bit about Natalia. She is a Mexican paleontologist working as the collections manager for the Invertebrate Paleontology collection at the University of Kansas. She completed her undergrad in biology at the National Autonomous University of Mexico and got her PhD from UC Davis, where she focused on combining analyses of brachiopod internal body structures and genetic diversity. And we had a lot of fun saying her talk title 7 times fast. Um, when we weren't live.
Long look at long loops. Long look at long loops. Long look. Okay, never mind. I had it earlier. I swear I had it
Yeah,
earlier.
he absolutely did, everybody. So, if any of you guys have questions for Natalia, as always, go ahead, put it in the chat. We will have a Q&A session after her presentation. I know I will probably have lots of questions because, again, brachiopods are cool. Don't know much about them. Natalia, whenever you are ready, go ahead, take it away, and tell us
Yep.
all about loopy brachiopods.
All right. Um, Perfect. So, Brittany already gave a little bit of a summarized version of my bio, but I wanted to talk a bit more about myself. So, I was born and raised in Mexico City. When I was a kid, I wasn't one of these kids that was super interested in paleontology or basically anything sciency. I um I like a lot of people that go into paleo as a career, I wasn't in love with dinosaurs. I wasn't in love with fossils. The most sciencey thing that I wanted to do maybe was being a marine biologist for a while there. I really like marine mammals, probably like most kids do. Then as I grew up and went to middle school and high school, sadly there's no awkward teen pictures of me. I grew up when film cameras and digital cameras, that weird transition. So thankfully there's no digitized evidence of my teens. But when I was a teen, I, I wanted to study chemistry, and I thought that I was going to go into like chemistry and do biochem. But eventually, when it was time for me to graduate and go to college, I wasn't really into it anymore. Um, so I kind of knew that I wanted to do something in science, but I really didn't know what. And the way that college works in Mexico is slightly different from how it works in the US, where you have to apply to college with a specific major in mind. So you have to make this big decision of really what you want to do for the next 4 years and possibly your whole career when you are 19 years old. Um, so it's like a big decision, and I was having trouble like figuring it out. And one day I was talking to my mom, and she asked me if I had ever thought about pursuing a major in biology. And I actually have never thought about it. I found it cool. I really liked animals. I thought that it might be actually a good option. especially because the curriculum of biology at UNAM really was very diverse. So you can go and do philosophy of science or biochem or molecular bio, genetics, or just do more traditional like systematics, taxonomy. So I was like, sure, why not? I'll apply to biology. I'll figure next steps later. So I started my undergrad in biology at UNAM, the National Autonomous University of Mexico. And another way that college, or at least the program of biology at UNAM, is different than how things work in the US, is that after your first 2 years, for the last 2 years of your undergrad, you have to choose a specific lab to work on and a lab where to do your undergrad thesis. Again, another difference, you have to do an undergrad thesis to graduate. So over the summer between that like second year and third year, there was a lot of talks from labs trying to recruit students for their labs. And I honestly had no idea what I wanted to do. I went to all of them: insects, genetics, molecular biology, and like really nothing was like speaking to me until I went to see the Invertebrate Paleontology talk. And I just found it like so fascinating. By that time, I've already had a course on paleobiology, and I really liked it. I really like the fieldwork aspect of it. So I was kind of like convinced. I was between that or neurobiology, but when I found out that I had to like work with live animals, I think they tend to like work with live mice at that point. I was like, yeah, no, probably not my thing. I'm more about dead animals in fossilized in rocks. So I decided to join the lab, and I did my undergrad dissertation there. And I did it on Cretaceous bivalves from the San Juan Raya Formation in central Mexico. So when I graduated from undergrad, I started my PhD directly from undergrad in UC Davis, working with Sandy Carlson, looking at brachiopods, which I'm going to be talking more about today. And once I graduated in the winter of 2019, I started a position as collection manager at the Biodiversity Institute at KU. All right, so after all that story of how I got where I am and how I never envisioned myself being a paleontologist as a kid or really any sort of scientist, I'm going to talk about brachiopods. So brachiopods are marine invertebrates. They have shells with two valves. They appeared in the early Cambrian, so around 542 million years ago. So basically, they have a really, really long geological history. And they have this thing called the lophophore, which is the feeding and respiratory organ. Brachiopods were highly diverse and abundant during the Paleozoic, which is here marked in pink. And after the end of the Paleozoic, during the Permo-Triassic extinction, they suffered a dramatic decline. So you can see how we have all of this diversity here. And then after that extinction happened, that extinction basically killed 80% of marine species. Then we have not really a lot of brachiopods. And that continues to be true right now in the modern. And the order that's more diverse today is the order Terebratulida, which is the one that I've been focusing on in my research. So this is probably like the main question that I get all the time as a brachiopod worker. Are brachiopod clams? The answer is absolutely not. They're not. They might be very similar in external morphology, but brachiopods are definitely not clams. They're not even mollusks. And a good way of distinguishing between brachiopods and bivalves is actually taking a look at their symmetry. So brachiopods have a dorsal and a ventral valve, meaning like up and down. And the axis of symmetry is basically perpendicular to the opening of the shell. Bivalves, on the other hand, have left and right valves. So the axis of symmetry is parallel to the opening. So in bivalves, we basically see that the two valves are exactly the same. In most cases, there's also some exceptions, for example, oysters, but in most cases, that's how it works. For all Pokemon lovers out there, here's a good analogy. Brachiopods are shelders, and bivalves are cloisters. So shelder has a dorsal valve and a ventral valve. And you can tell that they're symmetric if you cut a line perpendicular to the opening. So you have these two little protrusions in the shell. That's the same thing. But the ventral valve is not exactly the same as the dorsal valve. And then in cloisters, we have a right valve and a left valve, and the axis of symmetry would be parallel to the opening. So we have that the left side is exactly the same as the right side. So good analogy for Pokemon. But what's inside a brachiopod? So they might be similar externally, but internally they're completely different. Brachiopods have a lophophore, as I already mentioned. That's a feeding and respiratory organ. This lophophore basically creates currents inside of the mantle cavity that help feed the animal. Bivalves do not have a lophophore, and their internal anatomy is completely different. In good mollusk fashion, they have a foot and they have gills. So as you can see, they're completely different. And the next question that I usually get is, are brachiopods related to clams? And the answer is no, not closely at least. Brachiopods are part of a group called Lophotrochozoans, which means that they are related to phoronids, which we can see in the lower right side. Bryozoans on the upper image here. These two groups also have lophophores, as you can see in the picture. They don't have shells, but they do share that internal anatomy of the lophophore. Not as closely related to brachiopods as bryozoans or phoronids are annelids and nemertrians, which in like the most basic sense are worms. Why should we care about brachiopods? Well, they are definitely, as I showed in that little graph, they're one of the most abundant and diverse marine invertebrates in the whole fossil record. They appeared in the early Cambrian when animals first appeared, and they're still around today, meaning that we have all of this data that expands throughout the whole geological history of animals. And what can we learn from brachiopods? So given that we have modern specimens, these modern brachiopods can help us better understand the fossil record. And therefore studying these modern specimens will help us make better and more accurate inferences, for example, in taxonomy, systematics, ecology, and many other fields. So the order of brachiopods that I work with is called Terebratulida, and this is the most diverse group of living brachiopods. And this order is divided into two: Terebratulidina, which are they're commonly known as short loops, and Terebratulidina, which are the long loops. Here we can see in the upper right side, we can see a short loop, which basically are these, this structure right here. In this one is very short, and you can see in the long loops how it extends basically all the way in the shell. So what really are the long loops of brachiopods? So these structures are also known as brachidium. There are structures, they're calcified structures that support the lophophore. They are the most conspicuous and obvious morphological character in these brachiopods. Really, other than this, there is nothing inside of the shell. There's some muscle scars, there's some other stuff going on, but this is definitely like the most visible and the biggest structure inside of the shell. And they're very important for figuring out taxonomy and relationship between these species or genera. So they're very important phylogenetically. So how do we go about studying long loops? So as you can see from some of the images that I've shown, these long loops are very fragile, especially because these brachiopods are usually like a couple of inches in size. So the long loops are like very, very thin, very fragile, and they're rarely preserved in the fossil record. So the best way of studying long loops is by studying modern specimens. And how do we get access to modern brachiopods? Well, modern brachiopods are notoriously hard to collect. They usually live in very deep waters, and they're not easily accessible. So a good way of getting these specimens for research is to go to museum collections. These are some of the museum collections that I went to look for brachiopods, both modern and fossils. And it was great. It was great having the opportunity during my PhD to work at museums, to spend some time there looking at the collections, looking at brachiopods. And a lot of the specimens that I used for my research came from museums. Another way of collecting brachiopods is actually going on collecting trips, collecting expeditions. And I also got the chance to do that during my PhD. I went to Oregon, which was a complete failure because we didn't find anything, but it was a beautiful experience. I also went to Japan and to Monterey Bay. Here's some pictures. A very interesting experience, both of them. The one in Monterey Bay, I got to go on a cruise with the Imbari, the Monterey Bay Aquarium, and they deployed a remote operated vehicle, an ROV, to go to very deep ridges in the Monterey Bay Canyon, and it was incredible. It was incredible to see how they controlled the ROV, how they collected specimens, took measurements of. They were doing an experiment on sponge respiration. So it was a great experience that getting to see that. Also, the Japan trip was also very interesting. Instead of doing an ROV, we dredged. And we did find some brachiopods, very tiny, but they were great for doing genetic work. So once you get the specimens, once you have access to them, how do you go about studying them? So given that they have very complex shapes, the way that we decided to analyze them was by CT scanning the specimens. And what you basically get is something that looks like the brachiopod on the right. So once you CT scan the specimen, you create 3D reconstructions of the shells and their loops. And that's what I was interested in analyzing. So that the loop on its own, as you can see in like this picture. So the main question that I was interested in finding out is if there is a difference in shape depending on the species of brachiopod that I was looking at. So for example, here we have two species, Lacheus erythraeus and Lacheus vancouveriensis. They have very similar external morphologies. Sometimes it makes them hard to distinguish them from one another. They only vary a little on color and size. Lacheus erythraeus is definitely like the bigger one. Lacheus vancouveriensis tends to be smaller. So I wanted to see if there was a way of actually telling them apart only by looking at their loops. So that was the main, one of the main questions that I was interested in answering. And the way to analyze these 3D models was by implementing something called geometric morphometrics, which basically is an approach that compares points or landmarks, which are basically coordinates that are placed in the same structure across different specimens. And it helps to describe shape and quantify variation in shape. So for example, what we see rotating here is the landmark configuration for Lacheus. Here we had some independent points and some curves, and that's what we analyzed. And all of these points were selected for like each specimen and analyzed. Okay, so here are some of the results from that study. The main thing to look for in these plots is the way that the specimens cluster together according to color. So, for example, we see that all of the specimens here in purple, which is Lacheus rubellus here, cluster in morphospace. This is the name that we give these types of plots. And this is definitely a good indicator that you are actually seeing some real difference in shape. Here's some overlap, for example, between vancouveriensis and erythraeus. Like I said, they're closely related species. Sometimes difficult to distinguish. But they definitely, something I forgot to mention is that these two species definitely have like a not very, sometimes might overlap in geographic range, but they tend to be a bit different. So Lacheus vancouveriensis is found off the coast of Alaska, Washington, Oregon, maybe Northern California. And Erythraeus is found in Northern California, Monterey Bay, and Southern California. So yeah, basically the takeaway from this is that analyzing these statistically, we can tell that there is a difference between loop shape in each of these species, and that the classification accuracy for all of these, just based on long loops, is 100%. These are some plots for other species, Dallinella and Terebratulida, and it's the same thing. They do cluster by species, also showing us that loops are statistically different in all of these species. So, what did we learn from this from these analyses is that loops are informative characters, especially taxonomically, and that you can use these characters to reliably differentiate species. And then the obvious question really is, can this be done with fossils? So the answer to that is possibly can be done with fossils. Long loops are rarely preserved, like I already mentioned, but there's definitely some exceptional specimens like this one that we see here. That's the actual reconstruction for the loop for that specimen. We decided to take a bit of another approach to try to circumvent the fact that we usually don't find loops in the fossil record by looking at shell outlines. I really won't go into a lot of detail on that. You can read that in my latest paper. Just a plug for that. But this is something that can definitely be done, and it really demonstrates the power of CT scanning and 3D imaging and working with 3D models, and the amount of data that we can gather from this to analyze statistically and using geometric morphometric methods. So as a conclusion, brachiopods have a very long history on Earth. They're just fascinating. It's fascinating that they appeared almost at the beginning of when animals appeared, and they're still around today. That's great. That's great. We have that to inform us on the fossil record. And then long loops are an important morphological character that we can analyze and look at to help us understand how to classify species better. And thank you. Thank you for coming to my talk and listening about brachiopods.
Awesome. That was really
we
cool.
love listening about brachiopods.
Yeah, please tell us more. I really love the way that, like, well, first of all, your shelter cloister comparison just blew my mind. I was loving that.
Gabe and I live about an hour away from each other, and I swear I could hear just the scream of joy.
Good educational tool using Pokemon as
heck yeah.
analogies.
I absolutely love doing that kind of stuff. Brittany and I do cosplay for science where we dress up as Pokemon professors and talk about fossil Pokemon. So I'm all for that. So if you don't mind, I'm gonna kind of borrow yours and like
go for it.
Okay,
I'm sure I'm
cool.
not the first person to think about this.
That was great comparison.
Yeah, it was awesome. Okay, back on real science stuff though, but I absolutely love like how cool it was when you're able to CT scan things and see like such delicate structures. These are things that I thought like for a long time we thought wouldn't get preserved, but once you like really have the technology, it's amazing that you can see some of these like delicate structures that it feels like if you breathe on them wrong, they would break apart.
Oh, absolutely. We had a big lot of laquias from Monterey Bay in our lab at UC Davis, and I'm not proud, but I probably broke like one or two. Yeah, no, they're definitely very, very delicate. They're very thin. You breathe on them and they will snap. Yeah.
Oof, thank goodness for technology so that we don't have to do those again.
Blue.
And the other cool thing about CT scanning is that, so usually brachiopods, so bivalves are really easy to get the two parts of the shell open, but brachiopods are really, especially like these guys, they're really interlocked. So in order to open the shell, you have to break part of the hinge. So there's really no way of opening one of like Terebratulida or Laqueus without completely damaging the hinge. So another cool thing about CT scanning is that you don't have to even open them. You just put them in the scanner and it's perfect. And you also get the hinge in the CT scan.
Oh, that's so cool.
How come? So when brachiopods, they're not really like you said, they're not like bivalves. They don't really like open very much, right?
Well, they do open.
Oh, okay.
Yeah. I might be completely wrong, but my understanding is that bivalves are close together by ligaments. So it's like really easy to find just like disarticulated bivalve. But with brachiopods, you have to like break the shell.
Okay. Cool.
And given that there isn't like a huge amount of characters to study in brachiopods, especially again in these type of brachiopods. Breaking the hinge really, you're destroying a lot of valuable data.
Mhm.
Yeah. Oof. Thank goodness again for
Yeah,
technology.
we love technology.
Well, one more thing. Speaking of technology, how cool was that you got to be on that ship and got to use that or be part of that when they used the remote operated vehicle? What
Yeah.
was that like for you?
It was pretty wild. It was pretty wild because you're basically in the ship looking at all of their like screens, and they have like all of their controls there, and they just like send it off. The thing just like dives really, really deep. And you're just seeing how the thing just like goes down the water column for a really long time. Like it takes a while for it to even get to the bottom of the ocean. Um, and once there, like depends on where you are, but sometimes there's absolutely nothing. There's just like sand. And sometimes you just like see a lot of life. You see a lot of like sponges and sea stars and brachiopods, which I've never really, other than, I mean, you're still seeing it on a screen, right? But other than seeing some videos on YouTube, like I've never seen a real brachiopod hanging out in the bottom of the ocean, just doing its thing. So it's like pretty impressive, as especially because the other thing, like I entered this field and I entered all of these from a paleo perspective. So for me, brachiopods were just like the cool fossil, a cool rock. But then I kind of, moved a bit to like the modern side of things, and it's like, wow, no, they're actually animals. They actually have slimy bits inside. They you can actually like see them in the bottom of the ocean just hanging out. And that's just fascinating. It's fascinating to actually have the opportunity of working on a group that has such an incredible history and that you can still find them today. Because like trilobite workers don't get that.
Mhm.
Ammonite workers don't get that. Well, they can look at nautiluses, but it's not the same thing. So it was great working on brachiopods. I think they're a great group, and I think there's still like so much stuff that we can learn from them.
That's so cool. The little kid in me that wanted to be a marine biologist for a long time
Same.
ago just envies all that.
Uh-huh.
It's so awesome.
Is there like a Venn diagram between like kids who wanted to be a paleontologist and kids who wanted to be a marine biologist? Because I also, when I was a kid, I also was like, I'm going to be a zoologist, a marine biologist, and a paleontologist all at the same time.
And that's the thing. I never wanted to be a paleontologist. Like dinosaurs were not a thing for me. Like I remember having one book, and like that's it. Like they were cool, sure, but not even Jurassic Park did it for me.
Wow.
So yeah, right. I the most like sciencey thing, like I mentioned, was like wanting to be a marine biologist, which I guess I'm not that far from it, which
I
is
mean,
it's
pretty
impressive.
darn close.
Yeah.
Marine paleobiologist. That's probably like
There
what
you
you can
go.
call yourself to combine it all together.
Yeah,
Yeah, it's definitely it's
mix it all together.
a weird mix. But that's the
Alrighty.
thing about brachiopods. Like you can do that, and I think that's one of the main positives of working with brachiopods.
Heck yeah.
Alrighty. We have some cool questions from our chat. This first one is from Nathan, who asks, I'd be confused with which valve is up. Often people talk about the dorsal valves and ventral valves, but is that the actual life orientation for a brachiopod?
So, okay, at least for the ones that I study, they attach to the substrate with a pedicle. So there's no real like up down.
Mm.
I think it has more to do with development, like more than anything, and also and the difference between how do you tell what's the ventral and the dorsal? So the ventral has the pedicle, it has a pedicle opening, and the dorsal has the loop.
Oh.
Okay.
So if you find brachiopod shells, like fossilized brachiopod shells, you can probably guess based on that what valve you have. Sometimes the loop is preserved in like weird bits. Like you'll see like some evidence of it, even if you don't find the whole thing, which is kind of the problem with loops in the fossil record.
Cool. All right, I will
Noted.
try and remember that. Let's see. Do you have any ideas as to why brachiopods were replaced by bivalves after the Permian extinction?
Um, so I think really there isn't really like too much of an aspect of competition. They were just decimated by the extinction. Like yeah, bivalves actually take a lot of the ecological niches and do the things that brachiopods were doing, but I think that it is not a direct result of bivalve competition with brachiopods. They were just displaced to like weirder environments, deeper environments, environments with like less productivity, um, less light. Also, because like the metabolism of brachiopods is definitely like slower than bivalves, so that's why you tend to see brachiopods kind of displaced to these. Maybe displaced is a bad word because it's not a result specifically from bivalves competing with them.
Cool. Yeah, kind of just as a result of the extinction more than anything.
I would say so. Yeah.
Bivalves are just in the right place at the right time, I guess.
It seems that that's the case. Yeah.
All right. Our next question is from Sarah. Are fossil brachiopods useful in telling us things about the climate or environment where they were found?
Oh, absolutely. Especially if you so depends on the brachiopods. So there's brachiopods that were found in more like tropical waters, some that are definitely more deep water, marine. So for example, um, so you can also tell about the environment based on the assemblage of other species that they're found with. Um, so yeah, like that definitely that can be done. Brachiopods are great environmental indicators.
Do you think that they would be delicious? Do people eat brachiopods?
People eat brachiopods. It's a thing in Japan and in some other like Southeast Asian countries.
Oh,
Um,
okay. I
they
feel a little
usually,
I, I feel a little less bad for being like looking at your pictures and being like, man, that's making me a little bit hungry while you
oh
were
no,
presenting.
okay. So the ones that I worked on, they really don't have a lot of anything inside. They have like goopy bits, like they have the lophophore and they have other goopy bits. So they really don't have a lot to eat. But the ones that are usually eaten are the lingulids. So that's like the classic like living brachiopod, like living fossil brachiopod that has like a long tail, which is the pedicle, and that's what people usually eat. It's the meaty pedicle. I've never tried it. I would be up for trying it.
Me too. I think
Me
I
too,
think if I ever got
honestly.
the opportunity. I mean, you like oysters, Brittany, so maybe it's
I
kind of very
do.
similar.
I'm very into seafood and clams, so this is in addition to not having breakfast, this whole talk has just made me very hungry for some reason.
I think it's probably a bit chewy. If I have to guess the texture, I think it would be a bit chewy.
All right, we'll do a special episode one day in the future. We'll find some brachiopods and we'll just taste test it on the air and
Uh-huh.
we'll see how it goes.
Be
I'm
great.
I'm I'm down.
Okay.
Okay, cool.
Oh goodness. Um, this is another question from from Nathan, which I think is a good question. What's the weirdest or most surprising thing you found digging around in museum collections?
Oh.
Oh, in museum collections. So, one of my favorite things was at the Smithsonian. They have really big blocks of like reef forming brachiopods that they acid etched. So you basically see the brachiopods almost like they were in life, and they're very spiny and really cool. And I think that's like one of my favorite finds at a museum. Um,
Yes.
that was really cool. I love it. I love digging into collections.
Hmm.
Oh yeah. I mean,
And
I'm a
being
collections
a
manager
collection
too, so.
manager, like you can do that all the time.
Oh yeah. I mean, like, I do a lot more outreach now than collections management stuff, but like when I was doing some more of that, it was so fun just like opening a drawer and
Oh
just
yeah.
seeing something that like, hey, we have this? And then all of a sudden it starts this whole like research and story trail and you're just like, amazing. I love opening up cabinets.
Absolutely. Yeah, but that was one, yeah, that Smithsonian one was really one of my favorites. They have
Just,
incredible specimens, obviously.
yeah, that just sounds
Um,
so impressive. Oh my gosh.
I'm just gonna go ahead and answer the same question, but in real life collections.
Hmm.
My favorite thing was finding a sea spider from the bottom of the ocean in Japan. And it was the coolest thing because I've never seen one in real life.
Oh,
And they're it sounds dumb, but they're just like in the books or the internet, I guess.
so
They're super
cool.
cool. Yeah, and in, we found crinoids. I've never seen a real life crinoid. I was just blown away.
That is
It was
so
really cool.
cool.
Yeah.
I see all these
Uh,
things that, like, same. I've seen crinoids and brachiopods and whatnot in museum collections. I've seen them as an inert fossil, but to like go out
Yeah.
and find them in their actual environments today, that's so cool.
one
Yeah.
of those, the one thing that I would love to see, like in the wild by like any random chance, is a coelacanth. Like I know it's super hard to go and find them like in the deep ocean, but like if I could see one in real life, I would like be up on like the submarine glass, just like I love you, please.
Like, hello.
Aren't
Hi.
they like so surprising? Like I've seen them in museum collections. I've seen them actually in the LA County Museum.
Yeah,
Mhm.
And
That's where I've seen one
I was like,
and
wow.
smelt one.
Oh,
I don't think that would be delicious.
No.
why not though? They're just like fish.
It'd be greasy.
I don't know,
Huh.
maybe yeah, maybe I don't
Oh,
know. If I had the opportunity to taste it, I'll
yeah,
try
I
it.
guess I wouldn't say no. Um, let's see. Zoid has a couple of questions. Is it possible to grow bracts in laboratory conditions?
Ooh.
Yes, it is.
Really?
It's really hard from what I've heard. Um, you might end up killing a lot of brachiopods. But I think it's possible. Yeah, they require a lot of you know, their requirements are so specific that I think that's what makes it hard.
Mm.
But I know that they can be at least kept.
Huh.
Um, they had some in an aquarium in what's the name of this USC Marine Lab in Catalina?
Oh,
Oh, I don't know off the top of my head.
I can't remember, but I know
Well,
which one you're talking about.
I think they had some in Catalina Island in Southern California. Um, so yeah, it is possible,
Huh.
doable.
Did not know that.
It's like Subnautica. You have to have like the special like aquarium to like grow like the animals and stuff.
Very specific requirements.
Yeah, because and they don't like a lot of change, and the water needs to be very cold, and yeah,
Huh.
yeah.
Let's see. Another question is, do you have any opinion on the relationships of, I'm going to mispronounce this, Thessidata Thechideida. Could they be close relatives of Terebratulids? And could their lophophore support the answer?
Ooh, so their lophophore is kind of different than Terebratulids. It usually has, I wish I had like a picture to show, but it's usually sort of like lobate. It has like lobes, but they don't have like real support like these guys do. Like they don't have like the actual like long loop suspended inside of the shell. Um, um, oh, I'm not sure about that question. I I think, and this is from like conversations with my PhD advisor, Sandy Carlson, which she knows a lot about this. Um, she would be the real one to answer this question. Um, but I think they might be closely related to each other. Yeah.
I have no idea what that is actually. Is it another kind of brachiopod or?
Yeah, and they're very tiny. They are kind of tropical. And they're hard to find because they're tiny, and they are like they sometimes like are encrusting, so they encrust to rocks.
Interesting.
Yeah.
I'm gonna add that to my Wikipedia after the show, like I always do.
Yeah, I know. I read it and I was like, I have no idea what any of this is. This is fascinating.
Yeah, I'm not the best to answer questions about phylogenetic relationships and
That's okay. We always try to talk about, like, for folks at home, that like paleontologists or any scientist, you're like very specific a lot of times in what you study, and we try to answer questions, but sometimes it's like way beyond like what we've spent years like focusing on a very specific topic.
Yeah, it's kind of it's surprising how in depth you get into a very, very specific subject.
Yep. It's impossible for one person to know it all.
Oh, absolutely.
There's too much information out in this world of ours.
Yeah.
Yeah. Like when we used to do tours at the Alf Museum, and we'd have all these amazingly brilliant second graders and third graders come. They know so much more about dinosaurs than I ever do, that I'm just like, they're like, ask a question. I'm like, yeah, let's go with that.
Yeah. Even as somebody who is a vertebrate paleontologist, I guarantee you any 6-year-old probably knows more about dinosaurs than I do. I work on mammals. I'm like, dinosaurs are cool, but yeah, kids can teach me.
Oh yeah, no, don't ask me anything about dinosaurs. Absolutely not. And also doing outreach events, and someone's like, well, what about this vertebrate? Not even dinosaurs, like vertebrate. I'm like, where's the vertebrate student? Because I have no idea.
Well, we're hoping to change that with the show. Hopefully, we'll get more people who come over and ask about, like, hey, what about this brachiopod? And that will be like, oh yes, finally.
Finally, at last.
Yeah, and the only question is, are these clams? They're not.
Oh, Sarah said that the place is called the Wrigley Marine Science Center. I think that's right. Yeah.
Yes.
Yeah.
Yeah.
Awesome.
Correct.
Thanks, Sarah.
Thanks, Sarah.
Is there any more questions in the chat, Brittany?
Looks like that is it for today.
All right. Well, I've got a question that we usually am ending the show with. If anyone's interested in becoming a paleontologist or invertebrate paleontologist, what's some advice that you would give them?
I would say, I don't know if this is controversial, but I would say go the biology route. But either or works. Biology, geology, both of them are valid. Both of them will get you to the same place, because you will have to do graduate school. And it's a hard field. It's very competitive, but it's very rewarding. And there's so many things left to explore and questions to answer. So I would advise anyone that like is seriously thinking about pursuing paleontology to do it. Also, there's many skills that you will get in your career that will also let you do other stuff that not paleontology.
It's true. Very true.
Yeah.
Well, thank you so much for that, and thank you
Yes,
so much for joining us today and
yes,
teaching us
thank
all about
you.
these amazing
Yeah.
loopy brachiopods.
Thank you for having me.
Thanks so much to all of our audience too for joining us. Natalia, if people want to learn more about you or your research, where's the best place that they can find you at online?
Ooh, Google my name. It's kind of unique. Um, I have a Twitter. I don't use it that much. Uh, find me in the KU Biodiversity Natural History Museum website. Um, yeah, that's where I live on the internet.
Sweet. We'll try to put those in the description below. Um, and as usual, thank you all so much for joining us. If you like this program and want to support programs like it at the Alf Museum and the Western Science Center, you can find links on how to do that in the description below. And as always, make sure you like and subscribe to the channel for more stories from the world of paleontology. Thanks so much, everybody, and we'll see you next week because we have a special episode. It is Brittany's episode.
Gonna talk to y'all about some tiny horses.
All right. See y'all next week, everybody. Bye.
Bye.